Hi, I'm Z, and I'm a PhD student in the Brunton Lab at the University of Edinburgh, where we study stress physiology. Today, I'm going to talk about our recent paper published in the Journal of Neuroendocrinology, where we looked at the sex-dependent changes in neuroactive steroid concentrations in the rat brain following acute swim stress. Acute stress activates the hypothalamo-pituitary-adrenal axis, resulting in the production of glucocorticoids from adrenal glands into the bloodstream. Apart from the corticosterone, other neuroactive steroids such as allopregnanolone and TH-DOC have previously been shown to increase following acute swim stress in male rats in both the plasma and in the brain. Neuroactive steroids are steroids that can rapidly affect neurotransmission in the brain and can either be produced de novo in the brain or in the periphery and then enter the brain where they are neuroactive. Allopregnanolone and TH-DOC, for example, are allosteric modulators of GABA-A receptors, thereby playing an additional role in regulating the HPA axis via enhancing inhibitory neurotransmission. Sex differences are known to be present in the HPA axis responses to stress, where females generally mount a larger corticosteroid response, as shown here. However, it is not known whether the concentrations of other neurosteroids following stress are different between males and females and in the various different brain regions. In this study, we quantified a panel of neuroactive steroids using liquid chromatography, mass spectrometry, in the plasma and in 5 brain regions, namely the frontal cortex, brainstem, hippocampus, amygdala, and hypothalamus. There are 3 main takeaway points from this study. Firstly, we showed that there are indeed clear sex differences in the brain's steroid response to stress, with females typically showing greater concentrations of neurosteroids in most cases. Secondly, while concentrations of corticosterone, progesterone, and DOC were greater in both the plasma and in the brain, this was not the case for pregnenolone, DHP, DH-DOC, and allopregnanolone, as the increase was only present in the brain. This supports the concept of de novo synthesis of neurosteroids in the brain following stress. Lastly, we also found evidence for regional differences, especially for DH-DOC and allopregnanolone, which may be of functional relevance. Together, these data suggest that differential neurosteroid production may contribute to the sex differences in HPA axis responses to stress. To find out more, do spend some time reading our paper, and thank you for watching this podcast.